The Moon will have its own time zone. The White House said it had ordered NASA to develop a uniform time standard for the Moon. Politicians expect the creation of coordinated time that would apply, for example, to spaceships. These activities are aimed at increasing the safety of space missions. Time discrepancies may lead to errors in mapping and locating positions on the Moon or in its orbit. There is much less gravity on the Moon, so time passes a little faster there. The difference is small compared to Earth, but for new technologies, such as the GPS system or other complex computer and communication systems, it is of great importance. As NASA, private companies and space agencies around the world increasingly launch missions to the Moon, Mars and beyond, it is important that we establish celestial time standards, said White House official Steve Welby. A consistent definition of time among space operators is crucial for effective space operations, navigation and communications, he added. The Americans' goal is to create coordinated lunar time, CLT, without a unified lunar time standard, ensuring secure data transfers between spacecraft and synchronizing communications between Earth, satellites, future bases and astronauts will be extremely difficult. Timing discrepancies can also lead to errors in mapping and locating positions on the Moon or in its orbit. An atomic clock on the Moon will tick at a different frequency than on Earth. It makes sense that another celestial body, such as the Moon or Mars, would receive its own heartbeat, said NASA's Kevin Coggins, adding that everything on the Moon would run on accelerated lunar time. Why is this happening? It's all because of gravity. Time flows differently depending on where we measure it. Where gravity is high, time will pass slower than where gravity is weaker. Because there is less gravity on the Moon, time moves slightly faster there 58.7 microseconds per day, compared to Earth. When NASA last sent astronauts to the Moon, they wore watches to keep track of the time and compare it with clocks on Earth. But then precise timekeeping wasn't as important as it is today. These microseconds of difference matter when it comes to applying advanced technologies. The International Space Station, located in low Earth orbit, will continue to use Coordinated Universal Time, or UTC. However, NASA must determine, of course in consultation with other countries, where the new spacetime will begin. Coggins also admitted that, unlike Earth, the Moon will not observe daylight saving time. Scientists have detected traces of water in a distant galaxy. New observations made using the ALMA telescope in Chile have provided evidence of the existence of water in the most massive galaxy in the early universe. Researchers also discovered carbon monoxide in the galaxy SPTO31158, 12.88 billion light-years away from Earth. There are many water and carbon monoxide particles detected. This is the most detailed study of the gas content in a star-forming galaxy to date and the most distant detection of water in space. An article with the research results was published in the Astrophysical Journal. SPTO31158 is actually a two-galaxy system that was first observed by scientists at ALMA, Atacama Large Millimeter, Submillimeter Array, in 2017. Both formed when the universe was only 780 million years old. It was then that the first stars and galaxies were born. Scientists believe that the two galaxies may merge, and that the rapid formation of stars will not only consume existing gas within them, but could eventually transform them into massive elliptical galaxies like those seen in the closer universe. Using ALMA data, we detected both water and carbon monoxide molecules. Oxygen and carbon are first-generation elements, and in the molecular forms of carbon monoxide and water. They are necessary for the emergence of life as we know it, explains the lead author of the study Srivani Jaragula, an astronomer from the University of Illinois. This is the most massive galaxy that we observe at high redshift, i.e. at a time when the universe was still very young. It has more gas and dust compared to other galaxies from that period, which gives us many potential opportunities to observe particles and better understand how these elements influence the evolution of the cosmos, explains the scientist. Previous studies of galaxies have found a link between water and dust emissions, which can be seen in infrared light. The dust absorbs ultraviolet radiation from stars in the galaxy and re-emits it as far-infrared photons, explains Jaragula. This excites water molecules, which scientists can observe. In this case, it helped us detect water in this massive galaxy, he adds. Studying the first galaxies that formed in the universe helps scientists better understand the birth, growth and evolution of the cosmos and everything in it, including the solar system and Earth. Early galaxies create stars at a rate thousands of times faster than the Milky Way, says Jaragula. Research on the content of gas and dust provides us with information about their properties, such as the number of stars created, the rate at which gas is transformed, how galaxies interact with each other and much more, explains the scientist. According to Jaragul, there is still much left to discover about the galaxies of the early universe. Our study provides information about how far water is in space. But it also raises an important question. How did so much gas and dust gather to form stars and galaxies so long ago? The answer requires further research into similar star-forming galaxies, says the scientist. These exciting results demonstrate the power of ALMA, said Joe Pess, astrophysicist and ALMA program director at the National Science Foundation. These molecules, important for life on Earth, are formed quickly, and their observations give us insight into the fundamental processes of the evolution of the universe, emphasizes the researcher. fish released from aquariums threaten ecosystems. Giant specimens were found in the USA. The city of Burnsville, Minnesota, urged residents not to release unwanted pet fish into rivers and lakes. The appeal was made after a huge goldfish was found in one of the lakes. Species bred in homes and released into the wild can cause great damage to local ecosystems and threaten native species. The city of Burnsville shared photos showing several huge goldfish caught while surveying Lake Keller. Please do not release your pet goldfish into ponds and lakes. They grow larger than you think and contribute to poor water quality by destroying bottom sediments and plants, city officials wrote on social media. In Minnesota, goldfish are considered an invasive species that does not occur naturally in an area and can cause enormous economic and environmental damage. This also means that releasing them into public waters is illegal. Goldfish kept in a home aquarium usually grow to about 5 centimeters in length. However, they grow much larger in the wild and can be difficult to remove. They multiply quickly and can dominate native species. These fish are a long-lived species and in captivity they have sometimes lived to be 20 years or more. Residents of Burnsville had previously informed the city about the presence of goldfish in Lake Keller, so officials decided to conduct a study to assess their population. We expected to find goldfish in the lake, but was surprised by their size, said Daryl Jacobson, Natural Resources Manager for the city of Burnsville. According to Jacobson, goldfish tend to adapt to their environment, which is why they may be small in a home aquarium, but grow quite large when released into the wild. They are a tough species. They can survive under ice or in water with low oxygen content, he added. Wildlife researchers are dealing with a similar problem in nearby Carver County, where 50,000 were removed from waterways last October. Goldfish. This was part of a 3-year plan to study species that cause damage to ecosystems across the United States. But the US is not the only country that has a problem with free-roaming goldfish. In 2017, the City Council of Munich, Germany, reported schools of goldfish that were depriving all rivals of food in local ponds and lakes. The problem became so serious that the city threatened to fine anyone caught releasing pets into public waters. Huge goldfish have also been found in British waters. In 2010, a British teenager pulled a fish weighing 2.2 kilograms and measuring nearly 40 centimeters in length from a lake in Dorset. Canada and Australia also had problems with goldfish. First documented case of parthenogenesis in crocodiles. Zoo workers in Costa Rica reported the first case of virginity among crocodiles. A female American crocodile that had been kept in isolation for 16 years laid 14 eggs in her enclosure. Scientists suggest that dinosaurs may have also had similar abilities. A female American crocodile, Crocodilus acutus, living in Parque Reptilandia in Costa Rica, apparently got tired of waiting for a mate for 16 years and decided to cope on her own. Zoo employees discovered 14 eggs in her enclosure. Unfortunately, the final effect was not what the crocodile probably expected. The description and results of the research were published in the journal, Biology Letters. Parthenogenesis, also called virgin birth, is a process of reproduction that takes place without the participation of a male and involves the development of embryos in an egg cell without the participation of a sperm. This phenomenon is not that rare in the animal world, and in many species it occurs when there are no males around. This is how birds, some sharks, snakes and lizards, including large Komodo dragons, can reproduce. Now the first case of a captive female crocodile laying eggs has been reported. It took place at Parque Reptilandia in Costa Rica. The local crocodile, which had been living in isolation for the past 16 years, one day laid as many as 14 eggs. What's more, it turned out that half of them contained embryos. However, after 3 months of incubation, none of the eggs hatched. It was decided to examine the fetuses more thoroughly, including using a specialized program called parthenogenesis. What was obvious was confirmed, i.e., that the genetic material of the young that failed to hatch came only from their mother. Unfortunately, this case also exposes some weaknesses of parthenogenesis. Because the embryos created in this way are not always able to live on their own, or, as in this case, start this life at all. The case of the Costa Rican female crocodile led scientists to conclude that parthenogenesis abilities could be extended to the so-called archosaurs, i.e. primate reptiles. This is a group of evolutionarily advanced reptiles, which also includes crocodiles. Following this trail, we will come to the conclusion that dinosaurs and pterosaurs were probably also able to do this. Whether the young born through virginity will turn out to be healthy varies in different animals. For example, boa snakes and pythons seem healthy. However, not all snakes are so lucky. If we look at, for example, rattlesnakes, cobras, but also garter snakes and sea snakes, we will find that in their case such embryos are often dead or seriously deformed. Parthenogenesis is also possible in birds and sharks. But the health of their young ones also varies. Some of them actually turn out to be healthy. However, most of the observed cubs died prematurely anyway, surviving only a few months or at most years. The new organisms created in this way seem to be quite poorly adapted to normal life. By the way, it is worth confronting the view that virginity is a typical method of reproduction for animals kept in captivity. Well, this is not entirely true. Such cases have also been observed among animals living in the wild. Scientists have adapted stomach cells to produce insulin. Stem cells obtained from human stomach tissue can be reprogrammed into cells resembling insulin-secreting pancreatic cells, known as beta cells. Scientists have shown in experiments that transplanting small groups of such cells reversed the symptoms of diabetes in mice. Insulin is a hormone that regulates blood glucose levels. Without it, blood glucose levels become too high, which leads to diabetes and its numerous complications. High glucose levels are caused by a defect in the functioning of the pancreatic beta cells, which produce insulin. In type 1 diabetes, beta cells are destroyed by the immune system. As a result, the ability to produce insulin disappears. In type 2 diabetes, tissue sensitivity to insulin is reduced. As a result, the body demands increased insulin production, which exceeds the capabilities of beta cells. As a result, beta cells do not function properly and the level of insulin in the body decreases, although it is still produced. Type 2 diabetes is the most common form of diabetes. In a study published in Nature Cell Biology, scientists from Weill Cornell Medicine in the US showed that human stomach stem cells can be directly transformed, with striking efficiency, into cells similar to pancreatic beta cells. Scientists managed to take stomach stem cells and modify them to produce insulin in response to increased sugar levels. In typical circumstances, insulin secretion is carried out by the beta cells of the pancreas. However, in people suffering from diabetes, they are either damaged or die out altogether, which obviously affects the condition of the body. Interestingly, at the embryonic stage, the cells of both the pancreas and stomach are adjacent to each other. So certain possibilities could be expected here. Moreover, the stomach has its own cells that are able to secrete hormones. Hence the idea to study stomach cells to find a potential therapy for diabetes. Additionally, by collecting such cells from a person suffering from diabetes, it was possible to eliminate the risk of rejection of structures based on them. While this all looks very promising in principle, for years scientists have not been able to reprogram stomach stem cells to be able to secrete insulin in response to rising sugar levels. This was achieved by activating 3 proteins responsible for gene expression. In further studies, the cells began to show sensitivity to glucose, which confirmed that the researchers were on the right track. Does this solution work? The researchers decided to check this by implanting the modified cells into rodents suffering from a mouse model of diabetes. First of all, they functioned exceptionally stably throughout the entire period of observation. And most importantly, they were able to not only stop the progression, but also reverse the effects of diabetes. There is no need to convince anyone that this can be a huge opportunity for diabetics. However, several challenges will still need to be faced. First of all, scientists emphasize the fact that there are some important differences between human and mouse stomach tissues that will need to be taken into account. In addition, it will be necessary to ensure that the new insulin-secreting tissues themselves become more resistant to attacks by the immune system. This is a proof of concept study and gives us a solid foundation for developing therapies for type 1 diabetes and type 2 diabetes based on patients' own cells, said study co-author Dr. Joe of Weill Cornell Medicine.